Welcome to another episode of Interviews with Experts. Today, it's my pleasure to introduce Dr. David Peck. David is currently the Director of Research and Education at Better Bee in New York. He received his doctorate at Cornell University in Ithaca, New York, where he studied with Dr. Thomas Seeley and is credited in Dr. Seeley's books. I invite you to listen as we talk about the devastating Varroa destructor mite, as well as the importance of applying scientific methods when considering new treatments or beekeeping equipment. I'm Frederick Dunn, and here's Dr. Peck.
Hi, Fred, and all of Fred's viewers. I'm very pleased to be able to join you today and looking forward to this chat.
Okay, so welcome, David, and thank you so much for being here and agreeing to talk with me today. And a lot of people may be thinking that this is going to be a retread of some of your great talks that I've already seen dealing with Varroa mites and things like that, which, by the way, a lot of people got their first exposure to you at the Hive Life Conference in Tennessee back in January, where you did an impromptu presentation. It was really, really good, by the way. But today we're just going to be a little conversational because I want people to know a little bit more about your background and who you are, because we want to know who the people are that are educating us and kind of what's going on, and your credentials are pretty solid. So one of the first things I want, we already said that you're Director of Research and Education at Better Bee, and I think you also said to me that yesterday you spent your day teaching a class.
Yep.
You want to tell me a little bit about what you're teaching and what kind of class that is?
Sure. So one of my roles at Better Bee, and I wear a lot of hats, and the more I'm here and the more I do, the more hats people keep giving me to put on. So I've got my fingers on a lot of different projects. But one of my primary focuses is teaching courses through Better Bee to beekeepers, both locally and around the world. And so the course that I was teaching yesterday is part of our core beekeeping curriculum, we call it. So there are two classes. There's the core course and the Core Plus course. So the core course is for somebody who's interested in bees, doesn't know anything about them, and just wants to sort of build a good foundation that they can then continue to add to over the course of their time as a beekeeper. And one of the very first slides that I show in those lectures is, I can't teach you everything you need to know about beekeeping in the 6 hours that we're going to be together in this class. But what I can do is give you a good understanding of bee biology, give you a good understanding of tools and equipment that a beekeeper uses. And then we can talk about the most important skills: doing inspections, identifying disease, getting help when you don't know what you're looking at. And so we have that core course, which is for folks who don't have bees. We also have what we call the Core Plus course, which is actually basically the same class, but it's designed for people who have had bees for a couple of years, typically people who have had bees and lost bees and then bought new bees and then lost them again. And they wish that they had gotten that good foundation right at the get-go. And so it lets us go back, and I have people who are a little more experienced. They can probably find a queen or recognize a drone, but they don't know that much about the life of a queen. Often you find folks who have been keeping bees for 3, 4, 5 years who don't know that queens only mate once in their lives, or that they mate on the wing, far away from the hive and then return. And so it's this basic knowledge that you don't get if you just jump in and buy a kit or flip through a book and throw a nuke into a hive and hope that everything is going to work out for the best. Mm-hmm. So that's one of the goals at Better Bee in general, is that we sell bees, we sell hive equipment, we sell tools. But I don't want to sell someone something and then watch them fail as a beekeeper. I want them to succeed. And that's, partially selfishly, I guess, because if somebody buys bees from me and then they succeed, then they're going to be buying more hives next year when they decide they're going to split to prevent swarming. If somebody buys bees from me and they all die, then maybe they'll buy bees from me the next year. But if they all die again, odds are pretty good that they're just going to drop out of beekeeping entirely. And so that's really why education is such an important focus at the company, and why they hired me to come in and be in charge of these educational programs.
And that's a very good segue, by the way, into my next question, which is, of course, how they found out about you and where you were when they hired you for that job. But hold the phone, because I also want to know, when you're giving these classes, are they virtual or are they physical? Do people get to come there?
So right now, the classes have all been virtual, with the exception of some in-hive classes that we teach when the weather's a little bit nicer. We are finally able to find ways to safely start offering in-person classes again, which is something Better Bee's done for years that I haven't been able to do yet. But we've got a classroom, we've got space, and as we continue to monitor COVID and work with other folks to make sure that everyone feels safe doing so, we're offering in-person classes. But a nice thing that we discovered when I came in and started teaching these classes online was that Betterbee's courses, which for a long time had typically attracted folks only within sort of northeastern Pennsylvania, eastern New York, Vermont, in our local area, suddenly was drawing in students from Quebec, from Hawaii, from Florida, from Texas. And I've been delighted to be able to work with them and talk to them about their bees, because the nice thing is the bee biology doesn't change. The seasons do. When my spring starts or Florida's spring starts is different, but bees are still bees. So it's been really nice being able to reach people in a much broader way. And those virtual classes will continue even when we start up the in-person classes again.
That's outstanding. And that's kind of new for Betterbee to expand their education department? Was it kind of static for a while before you came on board, or?
Well, it's always been a priority of the company to educate, even from the very founding.
Okay.
And then when the next batch of owners sort of bought it from the family that had originally founded the company, the veterinarians that really have helped to expand Betterbee as much as it has, they also, they were veterinarians. They appreciated good scientific beekeeping and scientific understanding, and they wanted to spread that and propagate it. So those classes have been offered, but it might be a couple of classes a season. They by hiring me as the director of research and education, they've really solidified their commitment to, whatever salary they're paying me, that's their commitment to making sure that education happens. And the nice thing about my role is that no one ever comes to me and says, hey, I'm glad you taught those classes, but you should have sold more beginner kits or more books or something like that. That's not the goal of this job. And so as long as I'm teaching people, as long as we're getting positive feedback, as long as folks are enthusiastically calling and saying, hey, I took a class with David last year, but I really want to ask him a question this spring before I order X, Y, and Z. That's the kind of relationship we're trying to build. And so it's nice that I'm able to do that.
And something else that's very cool about that is often when you take a class for beginner beekeeping or the basics or even honeybee biology and things like that, you get in the classroom and you find out it's being taught by someone who either works for the store or something like that, maybe a local beekeeping member. Very rare to get someone who has a PhD related to this content coming into the classroom and talking to you. So I can't imagine somebody more qualified to teach about bees. It must really be, that would be part of the draw, is that you've got a real expert with academics behind them as well as the practical experience, and really provide endless resources as far as the education goes to those people. So I think that's fantastic. And that's also going to lead me into, how did they find you and how did they get you away from Cornell?
Well, my doctoral work at Cornell finished up. I got the doctorate. My wife was finishing up her doctorate at Cornell. And so I needed to stick around for at least a year. And so I was working in sort of side projects, a few little research projects. I wound up collaborating with the beekeepers in Newfoundland who are Varroa free and helping them to create a plan to keep the island of Newfoundland Varroa free. I also spent 3 months in Madagascar going and helping a nonprofit who was expanding a beekeeping program there because Varroa had only recently arrived on the island. And so their native race of honeybees were now in serious jeopardy, and the local beekeepers had no training or knowledge or resources to actually cope with it. So I did that for a while. And then my wife and I both applied for and got postdoctoral fellowships at Cornell, which were really focused on teaching. And that was because both of us recognized that we really like research, we appreciate research, it's so valuable to be trained in it and be able to do it, but we also really liked teaching more than many other professors seem to, certainly at some big universities. And so we wanted to expand that side of ourselves before we applied for professorships. And while we were teaching those assisting with courses and teaching courses through those programs, I was invited to give a talk to the Massachusetts Beekeeping Association. So I gave an online talk to them during COVID, and it was attended by one of the owners of Betterbee, Chris Cripps. And he was there, I think, maybe giving some brief spiel, or maybe they sponsored it, and so he gave a little commercial or something. But then he listened to my talk on Varroa biology, and he thought, wow, this guy's pretty bright. And so it was a few months later that they put out an ad for a director of education. And they really weren't expecting anyone with a doctorate to apply. They were expecting maybe someone who had an undergraduate degree in entomology, who would come in, could help them create a few classes and make a few slideshows. But when I applied, they really, to their great credit, were quite flexible about what it was they were looking for. I said, here's who I am. Here are my credentials. Here's what I'm interested in. Here's what I think I can offer. Do you think that we can make that work? And they were very happy to find a way to bring me on board. And so it's been really valuable to be able to come in, not just as, a sort of a focused class developer, but really as, a product development assistant. I'm working in product development and product testing. I'm teaching classes, I'm editing the newsletter, I'm writing content that gets printed into the catalog. I'm giving lectures, and I'm encouraged to go off and give lectures to teacher groups or student groups or garden clubs or whoever. And so the flexibility has sort of turned me into an academic in residence. It's not unlike if they just plucked a professor and said, instead of being an extension officer, for Cornell, why don't you come be an extension officer for Better Bee? And so it's been a really great opportunity to allow me to fill a void that I had already felt as a beekeeper and as a bee researcher. Because there are a lot of folks at universities who are being paid salaries to study honeybees, and there are a lot of folks who are interested in learning more about beekeeping, but the information that those researchers are collecting and publishing isn't necessarily the information those beekeepers are interested in. And so I had already started to see that gap. And I think moving into this role has made it even more obvious to me when beekeepers are calling me and saying, I wish someone would do more research on small hive beetles. I wish that there was a better way to control them. I wish that you could give me better guidance on what I'm supposed to do so I can fuel the spring buildup of my bees, but not be fueling the buildup of these beetles as well. And then to contact a lot of the academic researchers, and I understand why it happens to them, but to talk to them and hear them talking about how they need to get another $2 million grant so that they can study the genomics of some bacterium living in a small hive beetle's guts. It's scientifically interesting, it's scientifically valuable, but it's not that valuable for apiculture. And so I think a lot of beekeepers get left behind because the researchers are going off in their direction and the beekeepers are sitting here saying, I really wish you could just give me a better question about which queens I should be buying this year. Or rather a better answer about which queens to buy this year. And so I've relished the opportunity to jump in and try to bridge that gap a little bit.
That's really fantastic, because you really are now a jack of all trades, from course curriculum model management to research and development, and you're willing to go right in the classroom and talk to the ordinary people. I think that's
it's hard to get bored in my job.
Now, is there an area, if you had to pick an area based on that profile of all the different jobs, what's your favorite part of that?
Oh gosh. I'll be honest, I think my favorite part of the day is when I get a call from somebody in the customer service office and they say, somebody's asking questions. I've had bees for a couple of years and I can normally answer them, but this guy's going way over my head. Do you think you could talk to him? And they transfer the call over to me. And, sometimes it's a 5-minute conversation and I'm just answering a question. I'm just giving a definitive answer about the treatment window that you're supposed to be using for this miticide. But sometimes it's a 30-minute conversation where somebody's telling me their whole beekeeping story. They're asking one question and then I give an answer, and that inspires a second question and a third one and a fourth one. And so I honestly think that's probably my favorite part of it, is just talking to everyday beekeepers who have an interesting puzzle for me, who call and say, I've got this, but I've got a neighbor next door and he's got 10,000 yellow jacket colonies and refuses to spray them. So what would you do if you had more yellow jackets than anyone ever has had in the history of beekeeping? And I go, how would I solve that problem? And try to give advice.
That's really interesting. And I'm glad that you put that out, because we'll definitely publish your number down in the video description. Anybody that wants a free one-on-one consult with Dr. Peck, this is that's not.
No, you're more or less right. It does get in the way of.
Don't sign up for the class. Just call.
It gets in the way of curriculum development sometimes. But yeah, if you call and you stump everybody else in the office, the odds are good that you're going to get.
Yeah, you just happen to catch
me or someone else.
Okay, well, that's really good though. And the other thing is, Thomas Seeley wrote several books that most people are familiar with, and you're mentioned in a lot of them, by the way.
What I appreciate about Tom is that he gives credit to the folks who have assisted him in a lot of his research.
Mhm.
And it's always assistance, because it's his brilliant mind is the one that cracks a lot of these codes and solves a lot of these puzzles. But he does good research. He takes good notes. He communicates the findings very well, and he tells the story of the research. And I think an important part of his storytelling is the characters involved. And so he's a recurring character, but his grad students, his undergraduates who have assisted him, his postdocs,
Mhm.
are all folks that he both gives credit to, but also it helps to sort of color the story of the research.
Mhm. And is it true that you were his last PhD candidate?
I am the final minted PhD to leave the Seeley lab before his retirement. So I guess there's
And so-
always a possibility he could co-advise somebody as an emeritus professor, but I don't think so.
Yeah, and so he's retired now?
Yes, I think he's moved into full retirement, and he's doing. You asked me what my favorite part of the job was. If you ask Tom, it would not be teaching classes, although he doesn't mind doing it. It would certainly not be serving on university committees. It would be sitting in the bee lab, looking at bees in observation hives, conducting studies, and then writing books and writing them up.
Mhm.
And so that's what he's doing in retirement. It turns out that they never had to pay him to do that, because he would have done it for free. And so
Wow.
he's living, I think, largely up in Maine with his wife. They have a house up there, and he's conducting experiments and working on more books.
That's fantastic, and I hope he does keep writing books. And I took my Master Beekeeper program there at Cornell, and we had to give these final presentations. And I was really annoyed by a Master Beekeeper candidate there who talked Dr. Seeley into walking around with him to do his final project. I couldn't believe he volunteered to do that with a Master Beekeeper candidate. And I just said, wait, we could have called Dr. Seeley and gotten him to look to, and it was his thing failed because they were trying to do the following the wild bees, but in the Arnott forest. And of course they flew up and over the canopy of the trees and you couldn't trick them. But he gave his time to that student. I thought that was really, really interesting.
Well, that's the kind of scientist he is, and it's the kind of scientist he trained me to be. If someone asks for help and you're not otherwise occupied, go ahead and give it.
That's fantastic, and that's actually why you're here giving me this interview right now. I think that's really
right,
nice, David. Thank you.
I could be doing other things on a Sunday afternoon, but
yeah, you
could
be doing something else, sitting out in the snow here where I am. But you mentioned the Bee Lab. Tell me more about that.
The Bee Lab at Cornell?
Yeah.
Well, it's a little complicated, because the main honeybee lab at Cornell is the Dice Laboratory for Honeybee Studies, which I may have rebranded the Dice Laboratory for Pollinator Studies, because they do a little bumblebee work there as well. That's currently headed by Scott McCart, but it was founded many years ago as a honeybee research facility, and there have been a number of folks who have led it. Maybe the largest name, or the most name with the biggest impact in recent memory, is Roger Morse. And so he mentored a huge number of students. Many of the beekeeping or honeybee professors around the world that you might encounter either trained with him or trained with someone who trained with him. And Tom actually got his start in large part in the Dice Laboratory as a high school student. His father was a Cornell professor, and so he lived in Ithaca, and he was going in, he was fascinated by the bees, he was helping the graduate students. And when he went, I believe his story is that he, after his PhD at Harvard, he wound up at a professorship in Yale. And so he taught at Yale for a while, and then had this opportunity to move back to Cornell and join the Department of Neurobiology and Behavior, which was his department, my department. Not to be confused with the entomology department, which was the one in charge of the Dice Lab. So for a long period of time, there was actually the Seeley Honeybee Lab on Fries Road, and then the Dice Honeybee Lab on Fries Road in Ithaca. And so a lot of packages had to be exchanged between those two, because I don't think the post office ever really figured it out. But the honeybee lab that Tom ran was focused on animal behavior, honeybee behavior. And so we were asking specific questions about dispersal, about robbing, about disease transmission, those sorts of things. The apiculture lab, the research that took place at Dice, was really intended to be funded and by and for the beekeepers of New York. And so it receives sort of taxpayer money that helps to support it. It also is supported by private research grants. But all of the work that the DICE lab does is meant to communicate about bees to people, to do research that will be relevant to the beekeepers in New York and further afield. And so it really is an admirable organization, and it had sort of backslid a bit. It wasn't terribly productive for a while there. But I would say that the new Master Beekeeper program, a lot of the other educational programs and the tech transfer team that they're now operating out of there have been really valuable to beekeepers in New York and beyond.
Mm-hmm. So do you continue to collaborate with people at Cornell in your current?
My official status with Cornell right now is that I am an affiliate of the Veterinary College, which is yet another honeybee program. There's a honeybee health program meant to teach veterinary students and current veterinarians about honeybee health, because now that you need to consult a veterinarian in order to get a prescription to treat European foul brood for antibiotics, there's a requirement that veterinarians have some understanding of bees and bee biology. And so I recently wrote a chapter on varroa mites that is in one of these new textbooks intended for veterinarians to learn more about bee biology. And so I'm part of that program, and I go out, I think typically every year for some kind of a training workshop or a course that they're offering to veterinary students. So I'm affiliated with them, but of course I was just at the Mass Bee Conference in Eastern Massachusetts last weekend and hanging out with Scott McArt, who's the head of the DICE lab at Cornell. And so, he and I certainly communicate plenty. So I think that the formal connections between me and Cornell are not quite as extensive as the informal connections.
That's really interesting. And how did you end up there yourself? What was your pathway? How did you decide that you wanted to do what you're currently doing?
I've always been fascinated by animals. I've always been fascinated by nature. And so if left to my own devices with nothing else to do in a strange city, I'll probably wind up at the zoo. And so as someone applying for colleges, I wound up going to Tufts because they had a good biology program and a good psychology program and a good biopsychology program, really studying the way that brains and behavior interact with one another. And so I studied that for years. I also got part of my degree was in environmental studies. And so that led to me being exposed to a lot of behavioral biology. And one of my mentors while I was at Tufts was Phil Starks, who's a professor there. And he studies, among other things, honeybees. And he actually studied honeybees as a Cornell graduate student under Tom Seeley. And so sort of lazily, when I finished my undergraduate degree, I said, I want to go to grad school. Where should I go? And he said, well, I went to grad school at Cornell. It was pretty good. And I applied there, among other places. And I applied actually to work with Tom. I said, Tom, I want to come. I want to work with you. I'm really interested in honeybees, but I'm also interested in disease. And I know that there are a lot of disease issues in honeybees. And he said, you seem like a nice guy. You've got a good letter of reference from a former student of mine. But unfortunately, I don't like to take more than one student at once. I like to space my graduate students out a few years apart from one another, because I don't think I can give you a responsible and appropriate amount of mentorship if I'm trying to juggle too many people at once. And so he said, sorry, welcome to the department. I was accepted into actually another part of the program, but I couldn't join the honeybee lab. And so I asked him if I would be able to talk with him a little bit about beekeeping. Maybe I could get my own bees if they had any, spare splits to spare. And so I did a little bit of that with Tom, but otherwise he was just a friendly guy in the office next door, because he had another student, Michael Smith, who had come in and was working on, I think at that point, robbing behavior in bees. And so I did research on actually the behavior of ticks infected or not infected with Lyme disease, with the bacterium that causes Lyme, and studying the behavior of those ticks and how that affected their host selection, whether they were more or less interested in biting a mouse or a chipmunk or a possum or a deer. And so that project sort of ran its course. I wasn't getting very interesting results, and I wound up transitioning to another project studying chlamydia in mice, whether being infected with chlamydia would actually make a mouse more promiscuous. And so I spent a lot of time in a biosafety level 2 lab, going in, wearing my mask and my gloves and working with these mice and studying their mating behavior. And that also, it was an interesting project. We got some interesting results, but then ran out of funding. And so there I was with a lot of interest in disease, experience studying tick behavior.
Can I pause? Can I pause that? So what was the street level result of that mouse study? Did they become more promiscuous, or what happened to the mice?
The 10 set summary is yes, and it's interesting how. So there's all sorts of stories, and we'll get to this when we talk about the bees as well. I'm really interested in the way that parasites and hosts interact, and the way that their behaviors can change when they're interacting with one another. And so there's lots of fascinating biological stories about how a bacterium or a fungus or a parasite can get into an animal, get into its brain, secrete dopamine or eat up dopamine, and change the behavior of that animal. The chlamydia was doing something completely different. If you give a female mouse chlamydia, the odds are very good that it's going to cause inflammation that will cause her oviducts to get sealed shut. And so that mouse is going to be rendered infertile. What that means is that if I have a population of 20 female mice and as many males as I can find coming in to mate with them, if only one of those females has chlamydia, she's going to be infertile, and she's going to therefore never get pregnant, never stop reproductive cycling, never be focused on raising her pups, and will always be there cycling and mating with those males. And so it's a way that, essentially you can imagine that you can consider this a way that chlamydia could manipulate the behavior of that mouse. And it doesn't involve the brain at all. It's just getting in the way of normal reproduction, and then allowing that mouse to keep reproductively cycling and keep attracting more and more males to come and mate with her. So that was that. But this, to get back to my sort of main point there, is that I'm interested in parasite host interactions. And Tom, at that point, was really interested in this question of how the Arnott Forest bees were able to survive with varroa mites. And I said, mites and ticks are pretty close to each other, Tom, and I am still really interested in bees, and I don't have another project, and I don't have any funding to do anything else. And he said, well, Michael's moved far enough along, I guess I can accept you as a new graduate student. So I really started with Tom 2.5 years into my PhD, and then dove, wholeheartedly into the honeybee side of things. And so my sort of scientific story is not necessarily one all about honeybees. It's one all about disease, about parasites, about these interactions. And so for a long time, when folks asked me if I was a beekeeper, I would say, well, sort of. I'm a varroa keeper. I'm interested in these varroa mites because I want to understand their life and tell their story, not because I don't care about the bees, but because I think that the best way to fight varroa mites is to really understand them on their own level. And so I was interested in the varroa mites themselves and their interactions with the bees. And the best way to be a varroa mite keeper is to just be a crummy beekeeper. And so I had a few years in the bee lab where that's what I did. I had colonies where I was trying to breed mites. And so I just never treated. And there were plenty of other people doing experiments who didn't appreciate that. But it allowed me to get the parasites that I needed to actually do some of my own studies.
That's really interesting. There's a fundamental question I have about the varroa mite, and that is, because it depends on its host to survive, how does it benefit the varroa mite if it were to wipe out all the bees? Because I know that that's putting it in the plainest black and white terms, but you would think that you lose the host, you lose the parasite. So it wouldn't be in the parasite's best interest to kill off the species that it depends on because they're species specific. Is that right?
Right, they are. And it's unlikely that the varroa would be able to leap into yellow jackets or bumblebees if the last of the honeybees were dying out. The reality is, unfortunately, evolution isn't quite so smart as that. That organisms have no problem, especially organisms that cause disease or death, like the ones I study, have no problem hurting their hosts more and more and more over and over and over again. And even if that drives them to extinction, they'll continue to do it. The competition isn't between this theoretical varroa mite over here that keeps the bees alive and this varroa mite over here that keeps them healthy. It's these two varroa mites inside the same hive. Right? The one mutant mite that is nice is sitting right next to the mean varroa mite that reproduces 10 times as quickly, but it's going to cause that colony to collapse and die. So this varroa mite is never going to spread her genes. She's never going to get into the next colony and the next one and the next one when this colony dies and gets robbed out and the mites, go on their journey to the next victim. Because evolutionarily, she's fighting against these varroa mites that are just more competitive than she is.
Mm-hmm.
So it's the same thing that, when we look at COVID, it's nice to imagine that COVID's going to get nicer over time, but there's no reason to necessarily believe it will. Ebola doesn't get nicer over time. When Ebola gets into humans, it kills all of the humans in the local area, in the local village, in the local region. And then if it's not contained, it could kill every human on Earth if we let it. Typically, though, it burns out. It burns out so quickly that all of the people in that area either are not susceptible to it or they get it and die. And at the end of the day, there's no more, hosts for it to move into. Varroa, we because we aren't willing to let all of our bees die, Varroa always has new bees to move into. And so it can be as nasty as it wants to be, and it's just going to keep moving and moving and moving. That became a big part of my research during my doctorate. Tom brought me in to ask a very simple question. Well, not that simple. It took me a few years, but how are these bees in the Aranat forest surviving with Varroa? And so I was there testing hygienic behavior, testing grooming behavior, testing uncapping behaviors, all of these different things that we believe these bees are using to fight Varroa mites. But what became increasingly interesting to me is how are these mites spreading? How did they get into the forest in the first place? And if I killed every mite in this forest, how long would it take them to get back in? And that inspired a lot of my research on Varroa, really the behavior of the mites themselves, but also the behavior of bees that have Varroa mites on them. So we did research on drifting between honeybee colonies and how that spreads mites, robbing between honeybee colonies and how that spreads mites. And we even studied whether or not mites could spread off via flowers. And so that was a really interesting study. It was sort of the first, that was my gotcha moment for Tom, actually. So Tom Seeley and actually Michael Smith and I, so this other graduate student and I, all went off to the Cranberry Lake Field Station, which is owned by SUNY ESF. It's in the Adirondacks. And it's a place where Tom has frequently gone to do experiments where he needed to be completely, in control of all of the sugar that the bees had access to. It's nothing but pine trees, and so there's no forage. And so we went up there to do this experiment. And I think Tom thought it was sort of a joke. I think Michael thought it was sort of a joke. But the experiment was simply this. I had some papers in front of me written by Jeff Pettis, folks at the USDA, folks who had been in the US Customs and were inspecting shipments of cut flowers. There was one particular cut flower shipment from the Netherlands. I think there were tulips or something. They came in, they shook it over a screen, and they found this living mite. And they said, oh, I wonder what that is. And they looked at it and they passed it around. Eventually they took it to the bee lab and they said, that's a Varroa mite. That's a Varroa mite that is alive that was on those cut flowers. And so they published a paper where they said, we found this thing. It gave us some cause for concern. We thought, if a bee could go to a cut flower at a florist's stall out on a street corner and could pick up this mite and bring it in, that would be a way to spread mites, but also to spread whatever weird viruses this Dutch mite had on it. Maybe those viruses are in Europe, but not in the US. This is a way they can go. But they ended the paper by basically saying, but we have no idea if a mite could even do that, if a mite could even get onto a bee. So we went up to Cranberry Lake to do this experiment. And the experiment was simple. We did sugar rolls on some bees. We took the Varroa mites we collected and cleaned them with bottled water. And then we dried them off and set them on flowers. And we watched as bees came and visited those flowers, and we documented how many of the mites were able to jump onto them. And what we found was that, now I'm embarrassed to say I can't remember the numbers. I think we had 43 videotaped trials of 43 mites, and 41 of them got onto the bees and then flew off, or rather the bees flew off with the mites still attached to them back to the hive. And so it let us demonstrate, not that bees and mites are constantly, spreading parasites around out at these flowers. I don't think that that's what's happening. But what it let us do was demonstrate that these Varroa mites are not something to be taken lightly, that they are serious parasites that are seriously good at what they do. And so the thing that I said to Tom, and that I continued to repeat to him throughout my dissertation research whenever he doubted me, was never underestimate a parasite. That's a lesson I actually learned from ticks when I was studying them earlier in my career. Ticks feed on 3 different occasions in their lives, typically. A tick is going to feed as a larva, as a nymph, and as an adult. And they only feed once, and that's maybe for a period of a day or 2. Do you know how long a black-legged tick, a deer tick, lives?
I have no idea.
They can live more than a year. So
Oh.
most of a tick's life is spent living in leaf litter, crawling around on grass, trying to keep from getting dried out, not eating, not drinking blood, not bothering anything. It's just occasionally when they really need to grow and shed their skin that they jump up and get this big blood meal and then drop off and go through their next stage. And so, we think of them as these parasites that feed on us, but really, as far as they're concerned, they're just soil arthropods that spend a little bit of time drinking blood periodically throughout their lives. But a tick can sit and engage in what we call questing behavior, where they stick their 2 front legs out from whatever branch or grass or piece of leaf that they're attached to. And they'll sit there all day. And as the sun comes up or goes down and the humidity changes, they'll move themselves. But they just sit there waiting for a deer or a mouse or something to scurry past. The instant that host brushes past that leaf, that tick has to be on them. It has to do it perfectly, and it has to do it so subtly that a mouse doesn't immediately reach over and bite the tick's head off and throw it away. And they do it. They do it successfully all over the country, all over the world. And varroa are not to be underestimated in the exact same way. They have no eyes, but if I put a varroa mite on a daisy and a bee lands on the other side of that daisy, the varroa mite knows that a honeybee has arrived, and it does whatever it can to get there. I don't know where you got it, but I want one.
That was sent to me by a viewer.
Marvelous.
It's a 3D print, but yeah, it really does bring it in perspective when you see, and first of all, how agile they are. I've seen that video of the varroa on the flower and it clambered onto the bee. And then my question was looking at that, and I'm glad you explained the experiment too, that you sugar rolled it and you put the mites on there. How often do you think a varroa would leave its host when it's out on something like flowers or, when it's foraging? What's the incentive for that varroa to leave a guaranteed meal, basically?
Right. So that's the unpublished part 2 of that experiment. So the sort of the easy part was, if you put a mite on a flower, can it get onto the bees? And only when we proved that was it worth doing any more investigation. Your question is a very good one. Why would a mite ever be on a flower, and would a mite ever be on a flower? Could a mite jump off of a bee when it smelled floral nectar and then sit there and use that as a dispersal mechanism? Could the mites have evolved some protein in their brains that makes them recognize that the colony they live in is dying, and so it would make them more likely to do that? It's entirely possible, and I have no evidence that they do. What I do know is that if I walk around my apiary, I can go and find crab spiders and goldenrod ambush bugs that have caught and killed foraging honeybees, for probably half the summer. And I also know that if I take a varroa mite on a bee and I kill that bee and I pin that bee onto a flower as if it was being eaten by a crab spider, the varroa mites march off of the bees and they march out onto the petals of those flowers, and they stand there with their front legs sticking up in the air, sniffing, and I think waiting for a honeybee to happen past. Most of the mites that happens to are probably going to die, but if 1 or 2 of them are able to spread, that might be all it's required for them to get into Newfoundland or get into Australia. So from biosecurity perspective, just because you've got mites doesn't mean they couldn't bring in new viruses. And if you don't have mites, you need to be looking at the rare, acrobatic and impressive transmission modes, even if you know that that's not how most mites are spreading from colony to colony.
Yeah, because you only need one founderous mite to kick off the whole reproductive cycle. And
the horrifying reality of varroa is that because they reproduce by incestuously mating with their own siblings, because a single foundress goes into a single cell, has a male and a group of daughters, and they all mate incestuously, and because, just like honeybees, the males are haploid, they only have half as much DNA, they only have 1 copy of every gene. If a varroa mite ever had a mutation that made one of their genes faulty and it didn't work, well, the male would die. Or if they had genes that 2 copies caused the mites to die, well, then they wouldn't have been able to survive all of that inbreeding. And so their genome is basically really well cleaned up for surviving that inbreeding. It means that if I take one varroa mite and I put it on an entire continent, it can spread and there will be no inbreeding depression. There will be no risk of that inbreeding happening, because that's what they're built to do.
Mhm. That is really devastating information. So the other side of that is, you mentioned that the tick could live, for example, for a year without a host. How long does an adult Varroa destructor mite live without a host?
The longest I've gotten them to live in a lab was 9 days. And I think the longest published might be similar to that. It might be, 7, 8, or 9 days. So it's not that long. This is certainly not the way that mites are spreading. But if you've got a colony that is dwindling, you've got a colony that's dying. We have a paper that's currently under review on this very subject. A colony where the bee population is crashing. Maybe the queen has died. Now you've just got a few laying workers desperately trying to raise a few drones. As long as there are living bee bodies in that colony, then they are available to be robbed. Any honey that they've got is an attractive target for honeybee colonies nearby. And those varroa mites have something to feed upon. And on the day that the last bee dies, you could still have varroa mites that persist for, let's say, a week. And so that's a week with 0 guards, with a nice attractive honey odor leaving that hive. And now these varroa mites marching around looking for a honeybee to infest. So I think that robbing is probably a mechanism of transmission. It means that the sicker you make your host, if you're a varroa mite, the sicker I make my colony. If I kill them completely, as long as I don't do that in the middle of winter, it means that now I'm about to get a free ride into 3 different nearby honeybee colonies who are all going to send their robbers out to collect some of this honey and bring it back to their nests.
And that was going to lead me to my next question. Do they depend on the bees to maintain a warmth? In other words, how sensitive are the low end temperatures for varroa mites?
Varroa are sensitive to both temperature and humidity. And the disadvantage of living inside honeybee colonies, basically 100% of their lives, is that they don't have a huge temperature range, because the bees are so good at climate control. The varroa have sort of gotten used to that. However, honeybees go through winter as a very cold cluster. So they're keeping themselves warm enough that they don't freeze, but they're considerably colder than they are, when they're in the midst of brood rearing. And so the varroa mites can survive in a 50, 55 degree honeybee cluster in the winter. They can also survive in a 95 degree brood nest. And so they've got some range there. But yes, if you bring them outside of the bounds of what honeybees are typically going to be able to survive in, the varroa are likely to perish soon afterwards.
And does high humidity reduce their reproductive capability?
I've seen some data suggesting that high humidity is helpful and some suggesting that it's harmful. I've seen more data suggesting that heat during varroa reproduction excess heat is harmful for the mites, more so than it is for the bees. Chilling a developing bee and a developing mite will probably lead to major trouble for both of them. I've seen some data that suggests that if you heat up a comb or heat up a cell that has a mite and a bee developing inside of it, the mite's perhaps going to die or it will fail to reproduce properly, but the bee is probably going to be born with some sort of cognitive deficits. They don't develop properly either. Honeybees are spectacular climate control masters, and they do that because their brood need to be at the exact correct temperature and humidity at all times. So if you start messing with that, I think that yes, you might be able to interrupt the mites, but you're probably also going to interrupt the bees. And because they can ventilate, they can bring water in, they can, drink moisture, they can buzz their wings, they can bring in cold water and have it evaporate off of caps and then blow it away. Honeybees have a lot of ability to resist whatever temperature and humidity games you try to play with them. And so the odds are pretty high that your attempts are going to be foiled by the bees.
And I think Dr. Seeley did some research, or had an associate who did, where I think they even went to Hawaii and stress tested high temperature exposure to colonies of bees there with single entrances, and they managed to handle even those extremes. Do you know anything about that?
Yeah, not in much more detail than what you've described, but essentially, yes, that when you put honeybees through a challenge, as long as they have access to water and as long as you have enough bees, they can then control the temperature and humidity. It's just a question of how much labor they have to put into it, how much effort it requires. So they've got to burn calories for every trip that they take across the desert to go to the one little watering hole and bring back a one honey stomach full of water. But they'll continue to do that if they need to keep the brood cool. And so the capacity of bees to thermoregulate is really, I think, often underestimated.
So I'm going to have to ask this question because I know viewers and listeners are going to want to know the answer to it because I get it all the time. What about these thermal treatments for beehives that take the bees up to the limits of their, ability, but that will kill the mites, and then they bring these temperatures back down? Any thoughts about that?
If I was building a Varroa control device, I would certainly not build one whose goal was to increase the temperature of the brood nest in its entirety, because I know the bees will be working aggressively and probably successfully to resist me. If I built a device that was able to heat, if we think about a frame, if I was able to heat essentially the foundation and have that heat in the core of the brood, I don't know if the brood would fare terribly well. But I might be able to heat the mites that are underneath those caps, cause some problems for them before that gets out where the worker bees are, so that they begin their temperature regulation. So a device that was focused on the core of a frame, far separated from the workers, is probably, I suspect, going to be more successful than one that just sort of tries to heat the entire box, put a big sunroof on the top and let the sun pour in and heat everything up, or something like that. I haven't seen any data one way or the other on a lot of those devices, but as is often the case with Varroa control, if you heard about it 5 years ago and you haven't seen data proving that it works,
and
you might not see that data in the next 5 years either.
And I'm glad that you've said that, because that's what I always direct people to. Ask them for their published studies, and they are always in short supply. But this persists because the mindset is it's perfect, it's chemical free, it's just thermal based, and it takes hours, by the way, the stress. So, and I just want to bring it up.
I'll jump onto that and just say, one of the beautiful things about my job at Better Bee is that if someone comes to us and says, I've invented a Varroa control device, I've patented it, I've got 10,000 of them coming out of this factory that I've built, I'd like you to sell them. We aren't going to sell them because they're shiny. We aren't going to sell them because we think we can turn a profit. We're going to say, give us 10 and let us test them. And that's part of my job now, is coming in, doing that validation, doing those trials. And if it works, we'll publish it. And if it doesn't work, we'll probably publish that too. And so if you give us some things to try, you better make sure you've got a decent product. But if it doesn't work and you say, oh, but I could tweak the design this way and that way, we're happy to work with you. So it's that kind of scientific mindset that I really like at Better Bee. It's why I took the job, and I think it's something that I'm now able to sort of propagate and perpetuate. The idea that if you have a good idea, then let's test it. Let's do some science on it. Let's prove that it works. If you have an idea and you don't know how good it is, we can do science on it and figure out whether or not it was good. And if you have a bad idea, I don't want to carry it. I don't want to sell it. I don't want any of my beekeepers using it. And then I've got to call, they'll call me up and say, David, why didn't this work? And I'll say, well, because it doesn't work. Sorry, I sold it to you. That's a terrible conversation to have.
No, and I'm glad you said that too, because that shows the integrity of the company that you're working with. And I agree, which, by the way, is one of the reasons Better Bee is where I send more of my viewers than almost any other seller on the internet, just because.
Much appreciated.
Well, I know that's part of it. But what was your favorite field study that you've ever done, whether it was with Dr. Seeley, something that was really just the method and what you learned? And, do you have one that stands out?
I think, well, I have one that in some ways I really, really liked. And in some ways I wish there were 10 of me so that I could have done it 10 times. One of the papers that we published is called Mite Bombs or Robber Lures. And it's about this idea that maybe instead of talking about mite bombs, a single colony with a lot of mites that are sort of blasting the mites and the bees out to go infest everyone else, maybe we should be calling those colonies robber lures, because really maybe what's happening is that the bee population just sort of becomes pathetic and dwindles as the mite population skyrockets. And then they're attracting in all of the healthy colonies nearby to rob out the honey, and they pick up a bunch of mites. And so I thought it was a very elegant experiment. We just had a little apiary that had cordovan bees, bright, bright yellow bees. 100% of the bees in there were yellow. 1 meter to either side, we had New World carniolan bees. So there were black, black, black bees. 100% of them were black. 50 meters to the east and west and 300 meters to the east and west, we had more black, black, black bees. All of the black colonies I treated for mites, and so they had very, very few. The yellow colonies, I had been basically breeding mites in them as aggressively as I could all year. And so they were very much on their way to collapsing. And I put those colonies together, and then I went out and I just looked at how many yellow bees were in the black hive and how many black bees were in the yellow hive and what their behaviors were. And so I could start to figure out who was drifting when and who was robbing when. If I started seeing different colored bees fighting, that was a pretty good sign I had robbing. And so I think that was I thought a very straightforward and elegant test, and we published the results. I wish we'd been able to do it 50 times, because the more, the bigger the data set, obviously, the more confident you can be in your conclusions. But what we saw seemed to suggest that drift is happening. It's important. It happens the closer the bees are, the more drift you'll have, but it's happening even at long distances. But robbing seems like it's probably a better way to spread a lot of mites very quickly. I guess within that, my absolute favorite experimental method I used was one that I actually borrowed from Nickel Jacob, who was 1200, 1400s beekeeper. I can't even remember when it was from, but I think he published on bees in the 1400s. And one of the things he described was tracking bees back to their homes by blowing flour on them and then finding the flour covered bees as they went into whichever hives. And so I used a modified version of that by taking a plastic tube and some powdered sugar and just blasting the entrance to a colony that I knew was being robbed, and then sprinting as fast as I could to all of the other colonies nearby and figuring out where white colored bees were now returning home with ill-gotten gains. And so it let me demonstrate very clearly that not only were these yellow colonies full of mites being robbed, but they were being robbed by everybody. Every single one of those black colonies was sending robbers in. Those robbers were heading home, probably with varroa mites. Yeah.
That's interesting that you said that with the flour, because I did something last summer similar with open feeding, because I wanted to find out if am I open feeding my bees, or are they going to other, because really you never think of it. You just think, oh, they're close. My apiary's, 75 yards that way. These are going to be mine. So I put powdered sugar on them, and I got a special little sifter just for that. And I stopped open feeding because they were all going up over there. Because first of all, you could see them better against the blue sky. And I realized that I wasn't even feeding my own bees. Why am I even doing this?
So if you're the best game in town, I mean, a honeybee colony is more than anything else. I've already accused them of being a climate control system, but really what they are is an information system. They're a decision-making engine. And when a honeybee colony goes out and sends all of its scout bees out to visit all of the possible nectar sources, the whole dance language, the entire worker allocation algorithm these colonies are following allows them to very, very efficiently say, there are 50 different sources of sugar water in our environment. One of them is the best. So I'm going to dance intensely about it. I'm going to make you guys dance intensely about it. And eventually almost the entire foraging force is going to be going out and exploiting that high quality food reserve until it's depleted. And so if you're putting out, 2-to-1 sugar syrup in the middle of an open feeder, there's no flour in your habitat that can compete. So your hives, your neighbors' hives, even if your neighbors' hives are far, far away, if they send one scout and they discover that feeder, that's where all of the dances are going to be directed, and that's where all of the foragers are going to be until the feeder runs dry, and then they have to resort to the next best food, which is, maybe some goldenrod or some other plant nearby.
Yeah, there's a lot of opportunity for citizen testing, backyard testing there. Okay, I'm going to hit you up with some pronunciations. Are you ready?
Uh-oh.
Okay, here we go. And this is just because I get called out, shocking, I know, on how I pronounce different things. So I'm going to bounce it off of you. First of all, more than one larva. Is it larvae or larvae?
Larvae. I said larvae for a while, and then it was no less illustrious mind than Tom Seeley, who made fun of me in a lab meeting once and said, David, it's larvae. So larvae.
Okay, good, because I changed last summer. That's so I've been keeping bees since 2006 and 7.
Yep.
Last summer I learned to say that right. Okay, so the next one, pupae or pupae.
Pupae, same rule.
Dang it. I meant thank you. So anyway, and here's the other one. This people say that it really doesn't matter, but Webster says propolis, or is it propolis?
I say propolis. I don't know that either is correct or incorrect. And I do have to point out that I'm originally from Baltimore. And if you ever heard a Baltimore accent, you'll know that you really can't trust anything I say, because it is a funny talking place that I came from. Is that the last of your pronunciation questions?
Why? What are you gonna do?
How do you pronounce the active ingredient in Apogard or Apolife var?
Do you mean like thymol?
Yeah. So there are many people call it thymol. Many people call it thymol.
Right. I say thymol.
I agree. That's what I'm inclined to do, because it comes from the thyme plant. But the manufacturers of these products will sometimes say thymol to me. So I think it's like propolis. There's no real rule. Okay. It seems more sensible to call it.
Yeah.
Thymol.
Full disclosure. Why do I say it that way? Because Emma Mullins says it that way. And
I might have been the one who peer pressured her into switching.
So I wanted the best possible grade. So I pronounce things whatever the teacher is pronouncing. That's the way I say it.
Good. As a former professor and current educator, I encourage you to suck up your teachers whenever possible.
Absolutely. Yeah.
So, okay, now, and then I have a word of the day, because this is the one that I see mislabeled the most frequently. When you're looking inside the colony, the bees are making new comb on a foundationless frame, and they're all their little toes are connected. And they make this little string, this little gravity connection. I say that's festooning. What do you say that is?
I think my brain would call that festooning, although my brain would be willing to be proven wrong if someone came in and said, no, festooning requires that they be actively in a swarm or something like that.
So then what is festooning?
Yeah, yes, comb is built within a cluster of festooning bees, I think. So I think that's fair to say.
Okay. See, I'm using you now. That's what I'm doing. Okay, so the next, what else? Oh yeah, are you familiar with this app that people use on their phone? It's called Bee Scanning, for looking for varroa mites, phoretic mites on the bees themselves?
I'm not. When I teach people about varroa management, one of the things that I always do is I show them a photograph that I took from one of my experiments. And in that experiment, I had colonies that were absolutely devastated by varroa mites. They were totally overrun by parasites.
Yeah.
And in the photograph, these were bees living inside a glass-walled observation hive during the testing.
Mm-hmm.
In the photograph, you can see about 20 bees standing on the comb, walking around, and there are no varroa mites on them.
Right.
There are about 6 bees who are standing on the glass. And so you're looking at their undersides. And you can see that some of them have as many as 3 mites on one bee.
Mm-hmm.
And so they're riddled with these parasites. But all of the other bees, would appear to be parasite free. I suspect if we plucked them off the comb and looked at their undersides, you'd find just as many varroa mites. Mm-hmm. So when people say, oh yeah, I tested for varroa mites and that can't have been what killed my colony because I never saw any, that's a little red flag that goes off in my head. I say, what do you mean I never saw? Because if you're just looking at the backs of your bees, yeah, you might occasionally see a varroa mite, but you're not going to see them until the situation is already pretty dire. So if that's the system they're using, I'm comfortable saying they might want to revise their varroa framework.
Well, because here's the thing. When I said that, because if they're phoretic and if they're on the thorax of the bee, that means they're heavily infested because they're not feeding up there. They're feeding as you described. And but they said they also have factored that in. In other words, if you see one phoretic mite on the back of the bee, that indicates an exponentially larger population of mites on the underside of the bee.
I think there's too much stochasticity. I think that it's too random why a mite would occasionally meander across the thorax of a bee. And so I don't think that you could make I'm skeptical that they would be able to make a confident assertion that 2 mites on the thorax translates to exactly, 7,000 mites feeding on the undersides of those bees.
Yeah. So what
Could be. I'd love to see the data, but I'm skeptical until I do.
Okay. No, because it makes perfect sense. Because the other thing is, I always tell people who are using that app, and I've tested it myself. If you use that app and you get a treatable number of bees, looking at the backs of the bees, and that's definitely a colony you're going to treat, do not use it to say that your colony is clear, and you then will have to do some kind of mite count, sugar, or soap, and so on. So we're in agreement.
It's certainly anything like that until it's proven. You should always be backing up with a tried and true proven method, and making sure that things are valid, and you're not being misled by an app or misled by a new sexy test that's going to keep you from doing the appropriate treatments.
Right. And it also identifies brood disease. So.
Oh, interesting.
Yeah.
How?
By it recognizes anomalies in the surface of your brood when the bees are not covering it.
Okay.
So I see that expression on your face. You better watch it. So anyway,
I'd love to see the data. This is the easy thing about my job. No matter what somebody invents, no matter what they're selling, all I have to do is say the same thing. I'm a scientist, and therefore I'm skeptical, but I'm a scientist, so therefore I'm happy to be proven wrong. I'm a scientist. Give me some data that proves me wrong.
Yeah.
I love it. I love being proven wrong.
Healthy skepticism. That's And so then the next thing is, a lot of people this time of year are starting with their packages and things like that. And they don't have drawn comb, and they know that I've often recommended Better Comb to kick off a new package, for example. What is your recommended use for Better Comb? What's the most important niche that fills?
So there are 2 places where I would be most likely to reach for Better Comb. The first is if I had a package of bees or a nuke that I was getting set up, and I needed those bees to have enough space to begin their growth and begin their expansion. I didn't want to have to go through either the trouble or the expense, or most importantly, the time of feeding them a whole bunch of sugar water just so that they can build out natural beeswax comb. And what many beekeepers don't understand is how unbelievably costly that comb building is. That, by best estimates, it takes 8 pounds of honey to make 1 pound of beeswax. Well, that's 8 pounds of honey, but there's a lot fewer calories in a pound of thin sugar syrup than there is in honey. So it's not 8 pounds of syrup to make a pound of wax. It's 16 pounds, it's 20 pounds, it's 25 pounds of syrup to make 1 pound of beeswax. So it's a huge amount of feeding that you have to go through, and it's a lot of work that the bees have to go through to manufacture it. So if you want to save your bees from that trouble or get them growing faster, I would steer someone towards Better Comb. The other time that I'd reach for it would be if I had, and this is a problem that I have as a beekeeper sometimes, I might go out in late July and I might find a swarm. And I'll go, geez, that's a nice big swarm, but I don't have any spare comb to give them. And I know that if I brought them in, I could get them eating, I could get them laying eggs, I could get them storing away some syrup, and I could feed them and get them ready to go through the winter. But if I caught that July 31st swarm and then just threw them onto some foundation, there'd be no chance. They could build a comb, but then they'd die, and all I'd have at the end of the winter was comb. So it's again a resource I can give if I just don't have the time to give my bees to build all of the natural comb on their own. In terms of usage, it's the weird thing about Better Comb to the bees is that it doesn't smell like bees. So if I have a colony, if I have a nuke, or if I have a colony that's overwintered and they're in 10 deep frames, and then I take 10 more frames of Better Comb and put those on top, and I say, here's expand your brood nest, go ahead. They're going to look upstairs and say, that doesn't smell like home. That doesn't smell like anything. What a weird, attic that we've got. And they won't go up there. If you take sugar water and you spray that onto the Better Comb combs, then the bees are going to go up, they'll drink it up, they'll clean it up, they'll be tracking their little footprints all over there, they'll be sticking their bodies into the cells, and very quickly it absorbs that odor and it starts to it starts to be treated like natural comb that is part of their home. So spraying that sugar water, just any kind of syrup, thick or thin, I'd say probably thin, just squirted onto or even sort of dabbed onto it is going to make them more likely to walk onto the comb.
Mhm.
If you're starting with a package, you don't really need to worry about that, because if there's no natural comb for them to work with, then they'll just make do with what they've got. And very soon after a queen leaves a package and has an opportunity to start laying eggs, she's going to start putting them in the better comb, and then that's just what their nest smells like. But it doesn't have all of those bee odors because it isn't made from beeswax, and so they've got to sort of get over that hurdle before they're willing to start using it. The advantage, though, is that I can buy better comb, I can stick it out in my barn, and wax moths won't go near it. There's no smell to attract them. If I put a wax moth on better comb, it wouldn't know what to do. It probably wouldn't lay eggs, and it would just march off. And so, it's got that advantage that it's not going to attract small hive beetles or wax moths. It's also, if I put better comb into a swarm trap and into a bait hive, it doesn't have that same odor as really well-used brood comb that's going to attract bees to it. And so it's not like it can do everything. We call it better comb because we're better bee, but that doesn't mean it's better than natural comb. If I had natural comb, I'd probably use that. But for a colony that I really want to give a good early aggressive start to, or a colony that just doesn't have the time to build all of that comb, better comb is the thing I'd reach for. The other advantage is if you've got bees and you've got all the brood nest in the world, you're not buying packages because you're such a great beekeeper, you're just splitting your overwintered colonies year after year, you still might find yourself going out, checking the bees and realizing you've got every single honey super that you own out on those bees, and now they're bringing in nectar from some new plant. Suddenly the basswood trees are in bloom, and your bees have nowhere to store that nectar except down in the brood nest. If you don't want your bees to get honey bound, you've got to put a drawn super on top of them. But if you don't have drawn comb, then you can't do that. You can't just give them foundation and wish them luck. And so better comb is a great opportunity there to say, hey, I need 10 medium better combs shipped to my door, and they'll get there within the week. You can slap it on top of the colony, and now they can go right up in and start making honey instead of winding up honey bound underneath.
Okay, are you ready for this question?
Sure.
All right. When the colonists brought bees to this continent, were they bringing bees to this continent, or did the bees use humans to spread themselves throughout the world?
I think an interesting alternative to that is, were they simply returning honeybees to this continent?
What?
Because some of the oldest fossilized honeybees that we have in paleontology were actually found in North America. There was a native species of honeybee, some ancient cousin of all of the currently existent honeybees, that lived in the Americas. And so it's not like these flowers or these landscapes or this land, these rocks haven't had honeybees occasionally alighting upon them, in the past. It's just that they were gone for the last 20 million years or so. And so the return of honeybees to this continent, in some ways, yes, certainly it's not an invasion, because I haven't seen good evidence that they are a consistently invasive species that harms the ecosystem. But they are not native in the sense that these plants and animal communities had been living a very long time without honeybees. But I think that those honeybees are in some ways, well integrated and well naturalized into these ecosystems, because this is the kind of ecosystem that honeybees have lived in for a very long time. I don't know that the bees were telekinetically controlling the pilgrims, though, so I'll be inclined to go away from that.
Yeah, that's good. Okay. Is there anything new coming out at Better Bee that we should know about?
Let's see. We've recently added the HiveGate, which is a product that I know that you've taught many people about. You can probably grab one right there. Perfect. So we have recently added the HiveGate as a product that we are carrying and are excited to do so. That's another one of those examples of a product that came out. It was a gimmick. It was a gizmo. And so as skeptical scientific beekeeping suppliers, we didn't immediately say, give us 10,000 and we'll sell them to everyone who breathes. We said, does it work? What is it good for? How does it work? And so that's where we rely on folks like you and the community of beekeepers you've assembled to actually get these products, try them out, and as early adopters, either say, this thing's bunk, don't get it, or say, hey, this really seems like it's working. I really like what this is doing. It seems like it's delivering upon its promises. And so it's really based on your work and the work of other beekeepers we trust that we said, hey, this seems like it's got good potential. We are going to test it in our own hives this summer and try to figure out more about what it's good at, what it's bad at, what its quirks are. But we're very happy to be able to offer it and save people on the international shipping instead of getting it directly from New Zealand. We're also, another product that we're just about to add is, I'm trying to remember the name that we've given it because we didn't want to confuse people, but that means it's run through 7 or 8 different trial names. It is a, gosh, I can't remember the name, but it is a frame cage in which you put a single deep or medium frame. Yeah, maybe you've got one of them kicking around too. Similar in some ways, but it's a frame, which I suspect that is made out of queen excluder, but it's a cage in which you can simply take a single deep Langstroth frame, drop it down, put the lid on top, and now you've got a cage that the bees can comfortably walk into and out of, but that the queen can't enter, or more importantly, most of the time, leave. So if I have that queen on that frame, I put her down into that cage, I've limited and controlled where she can lay eggs. And I like it because this is something that I used a lot as a bee scientist. I would do experiments where I sometimes needed to have queens laying eggs because I needed pupae or larvae of exactly this age. But it's something that the beginning beekeeper probably has no business with. But there are a lot of things that a more advanced beekeeper or a more experimental beekeeper might find really valuable about it. The ability to put a queen directly onto a drone frame and have her fill that up with eggs so that if you're trying to do drone comb removal to trap varroa mites, you know exactly the day that the first and the last drone egg was laid. You can also put a queen in there if you're grafting and producing queens of your own. You can put that queen into the frame and now you know exactly the age of all of the larvae if you pull her out a day later. So I can go in and do my grafts and say, do I really want 2-day or 3-day or 4-day old, larvae, which are going to work better for me and the technique that I'm using. So it's got a lot of potential. You can isolate the queen, limit the amount of brood that the colony produces, which might be a good way to knock their brood numbers down, and then do something like an oxalic acid treatment or a Hopguard treatment that's better when you don't have a lot of brood. So there's a lot of manipulation and control that a beekeeper can get without the stress of taking a queen and just using a wire mesh push-in cage, limiting her to a tiny area, having all of the bees have a strong incentive to chew their way through it to release the queen. So I think it's going to be popular, and I'm really excited to see all of the different ways that some of our more advanced beekeepers make use of it, and then hopefully take photos and report back.
And you know what? That was unexpected. I'm really glad that you are going to carry that, because it was carried by another supplier and they just stopped carrying it. In fact, I bought a whole bunch of them from them and then shared with my viewers about it to create a brood break, for example, isolate the queen, and then be able to treat with OAV, and everything's phoretic at that point.
So many beekeepers and folks, beekeeping supply companies and also beekeepers themselves, come up with this idea, create something. Sometimes it's somebody like you in your shed making 30 of these a year and passing them around. And then if you have a heart attack and keel over or something, now nobody knows how to make it and there are no more to be had. Other times you have a company that goes out of business or gets bought by another company and they decide not to carry something. And all of a sudden a product that has been absolutely critical for somebody's, bee management is suddenly no longer available. And now yours breaks and you can't buy a new one. So those are the sorts of things that also, Better Bee is a company owned by beekeepers, run by beekeepers. If someone is hired and isn't a beekeeper, one of my first jobs is to say, look, you're a beekeeper now, you're going to be out in the apiary with me all summer. By the end of the next year, you're going to have bees at your house, or you're going to have some bees in our teaching apiary, because you've got to be a beekeeper too. And so what that means is that we feel the pain of beekeepers. If there's a product that we can't get because nobody can get it, we're going to be working hard to get one, because it's annoying us that we don't have one. And we're sure that it's probably annoying other people too.
No, but is it going to be metal like that one?
Yep. Yes, yeah, I've seen we've only got one prototype right now, but
Okay.
we're taking pre-orders, and it is beautiful. I used one. There were a whole batch of them. Maybe they're similar to yours that were in the Sealy lab when I was doing those experiments. And I actually like this new design better than the old one because it's easier to lift the lid right off and manipulate that frame without having to pull the whole thing out of the hive and manipulate it and then put it back in.
Okay, I'm gonna put in my purchase order because as soon as they're available, I want one. And also for those that are watching, when it's available, I'll put a link down in the video description here.
Sounds good.
So if you don't see it down there, you can't buy it yet. So that's pretty much wraps it up for today. Is there some closing advice you'd like to give? My primary viewers are backyard beekeepers. Do you have some general closing statement you'd like to make?
In order to succeed in beekeeping, you have to accept that you're never going to know everything there is to know about bees. And so as an educator, it would be great if I could say, take my 12 classes or, take this master beekeeper program, and that's going to be it. Once you've done that, you'll know everything and you'll be all set. The master beekeepers that I trust, whether they're taking the Cornell class or the Montana class, or they're, certified through EAS or other apicultural societies, the ones that I trust the most are the ones who say, yeah, I'm a master beekeeper, but that doesn't mean I don't make mistakes. It doesn't mean I don't screw up. It doesn't mean that I'm not sometimes confused about what my bees are doing. The professors that I trust the most, Tom Sealy, other bee researchers, are the first people to say, we don't really know everything there is to know about honeybees. We don't really understand everything it is that they're doing. There are still questions and mysteries in these bees. And so if you ever feel yourself with the attitude, I think I get this, that's the beginning of a failure for you as a beekeeper. If you feel like you're not quite following, if you feel like you're doing something, but you're not positive it's the right thing to do, that's where success is going to lie. And so you just need to accept sometimes you'll make mistakes because we don't know all the answers. But if you make those mistakes and learn from them, you won't make them again. And if you stack up enough of those lessons, and if you accept that you're always going to have to learn a little bit more in order to do a little bit better, I think you're going to become a better beekeeper over time. That's as much as any of us can ask for.
That is very well said. And I can say that, though, that was and is probably one of the greatest aspects of beekeeping, is that every bee season, we're looking forward to figuring out something new that we didn't know before, and then sharing it with everybody.
It would be so boring if I opened every hive and said, I know exactly what's happening. It's so fun to open a hive and to go, why is that happening?
Yeah, exactly. And hope to get it on camera, because I have my bucket list of things I've tried to get videos of. Although one of them is the mud dauber wasp that I want to get, because I've seen them fly into spider webs. The spider acts like it's stuck. The spider comes out to get the mud dauber. It grabs the spider.
Whip that abdomen around. Yep.
That is the best. And when you see it, it's like a miracle, and it really does happen. And it's like that with bees. If you've read a bunch of books, you've attended some classes, and you learn about all these behaviors, the first time you sit down and see a waggle dance, you get it. Or you see the tremble dance. This bee is trying to unload its cargo and nobody's paying attention. And all these things are, that's what that looks like. So that's great. I want to thank you so much for your time today. And again, for those watching, listening, this is also on Podbean, The Way to Bee. And the links will be down in the video description. Those will be updated when new things come out.
Terrific.
So, thanks a lot for being here.
It's been a pleasure, and thank you everybody for watching and paying attention to this man. And because he's got, occasionally he has some kooky ideas, but for the most part, he's a good, trustworthy source of information. And that's what you need to do: stitch together good, mostly trustworthy sources of information. And don't trust anybody with your life. Always be skeptical, but if you got somebody who tends not to lead you astray, keep listening to them.
Great, good advice. Thank you, David.
All right,
that's the end of it. And that's the end of another interview with experts. Don't forget to look in the video description for links and for more information and resources. Thank you for spending time watching. I'm Frederick Dunn, and I wish you all the best in beekeeping.